So, welcome everyone to um, today's biological approach revision session. And so, wait. So here's the things that we're gonna cover. And before we cover the biological approach, let's just review again the principles of biological approach. The first would be that the behavior is the product of physiology. What this means is that the structure and process of our brain and nervous system governs how we feel and think. So, um, behavior is made by, in biological approach, sometimes we assume that some behaviors are made by our brains. And the second would be behavior can be genetically inherited. So some behaviors may be passed from parents to offspring through genetic inheritance. And the last one would be that animal research may inform our understanding of human behavior. This means that some animals, such as monkeys, rats, and etc., have very similar brains to humans. So it's assumed that um, there are biological and behavior aspects that applies in animals and also in humans. Well, this is not always true. However, there are such links, and it will be explained later, unless if you're um, unless if you're not an eggshell. All right, so wait. Okay, so we will start with brain and behavior, and let's review the techniques to study the human brain. So there are many versions of this, but um, the version that I'll take is that there are three main techniques to study the human brain, which is autopsy, selective destruction, and brain imaging. The first is autopsies. So basically, in autopsies, you examine a corpse for structural differences between the norms. And what this means is that um, you examine you examine the brain of a corpse and you compare the shapes and, well, its freshness or how hard a corpse um, brain is, and you see, like, you observe physical structural differences. One example of this would be in Brugatan. It's a study where a guy observed about how there's this guy keeps on saying tan tan tan, and in the end, during the autopsy, they observed that a part of their brain was actually damaged. And then selective destruction means that incision into selected brain tissue and observing its results. One example of this would be the HM patient experiment. And keep in mind that these kind of experiments are usually highly unethical. However, sometimes in certain circumstances it is done. For example, in HM Milner, it's done because the patient had epilepsy, and and there was informed consent considering the incision or removal of this particular brain tissue. And brain imaging is one of the techniques to directly or indirectly image the structure of or function of the brain. And there are many kinds of this. But I'll just cover magnetic resonance imaging and functional resonance imaging because those are like the one of the two common ones that are used in psychology studies. Okay, so do you guys have any questions overall? All right, so since I see no question, oh. All right, so since I see no questions, overall, um, I'll just continue. And if you guys have any questions, feel free to put down any questions then right there in Discord, and I'll try to answer it. All right. So magnetic resonance imaging um, are MRI scanners, and as you can see in the picture, it's a big um, it's a big machine which uses strong magnetic fields, magnetic field gradients, and radio waves to generate images of the organs in the body. So, um, how how this machine works is that it uses um, magnets, magnetic fields, um, and bounces them into the nucleus or protons in the brain. And because every nuclei or proton has its own position, it has their own radio waves um, when the radio waves bounce back to the magnetic field. So um, the different radio waves which bounce back into the magnetic field creates an image of organs and tissues. And these radio signals are processed to show different body tissues. And examples of these um, examples of the studies which uses magnetic resonance imaging would be McGurr et al., Corkin 9117, Dragonsky et al., and many more. In here, I just simply put in the studies usually used in uh, the Oxford IB Psychology textbook or in Thinking, because I'm pretty sure there are like lots of options, but I'm just covering up examples of the studies you can use. Maybe if you have like other studies which uses MRI, it also works, but these are just examples. So magnetic resonance imaging. So every brain imaging scan would have their strengths and limitations. And for MRIs, the strengths would be it is very non-invasive and it has little to no risk of harm because you're using magnetic fields. And the good side of using these is that they have high resolution pictures since they're relying on nucleus or protons to bounce back radio waves. So by doing that, you can clearly detect which brain tissue is which. However, even if they are they have some strengths, they also have their own limitations, such as the fact that they don't indicate functional structural differences. So you can only see pictures, and you can't determine what function they might have. And in addition, you can't link it causatively to behaviors because there are no studies of like experimental studies which can be studied through MRIs. So another one would be functional magnetic resonance imaging, and this one measures brain activity by detecting blood flow changes. As you can see in the picture, um, when someone does something, different parts of the brain would have different blood flows due to like the activity that was done. And as you can see right there, they also do the same thing. They're similar to MRIs, but it's a further development of MRIs. And um, other than that, they also take multiple snapshots. So they would observe the regions with rich and low blood oxygen flow. And they do this so they can observe the functions of so they can observe like how fast or how slow the blood are flowing, which can show the activities on the parts of the brain. So the same as MRIs, they also use nucleus or protons to observe these differences. And examples of the studies that uses functional magnetic resonance imaging would be Baumgartner et al., which studies about which studies about oxytocin in breaches of trust. Harris and Fiske, 2006, the role of brain in prejudice. Sher et al., 2007, biological basis of flashbulb memory, and Fisher, Aaron, and Brown, 2005, dopamine on love. So the strengths of these studies would be that it is also non-invasive due to the nature of the machine. It also have little to no risk of harm. It is also easy to carry out, and it can causatively indicate links between thinking and brain activity because you can see the brain activity changes from the oxygen flow in the parts of the brain. However, when you do these kind of studies, there would be very unnatural. It would be a very unnatural setting because you're going to do an experiment with the machine right beside you, and as you can imagine that it would be very artificial and it wouldn't be ecologically valid because it's not how real life works. You don't do things right beside a machine. And um, I actually compiled like the some of the brain imaging functions, data gathered, and advantages and disadvantages, but I'm too lazy to put everything in because um, I doubt anyone would like mostly use this anyway. So like, if you guys actually want to use this, then just look at it in the presentation. Okay, so now we're jumping into localization. Wait, actually, before that, do you guys have any questions? No? Not at all? Okay. All right, so let's just proceed then. Um, so localization is the functions of specific parts of the brain and how they relate to behavior. So as you may know, behaviors are very complex, and several parts of the brain are usually involved when behaviors happen. But um, in psychology, there are several perspectives about localization. There is strict localization, there is the idea that specific functions are distributed, and there's also relative localization. The first would be strict localization. As you can see from the name, it means that specific parts of the brain have specific functions that are related to specific behaviors. An example of this would be McGuire et al. About the London taxi drivers. The second would be that specific functions are distributed. Basically, what this means is that, for example, there's a theory, according to Lashley, 1929, which says that memory is distributed, and it's also because of principle of mass action and equiponentiality. And basically, what this means is that it doesn't cover like the strict localization theory. And for example, for equiponentiality, it means like how when there's a damage in the brain, some of the other brain would try to recover that kind of damage by distributing the processing of some functions. And there's also the principle of mass action, which means that basically if your brain's gray area is bigger, that means that it has more it has like a bigger function than a better functioning than like a brain with small gray area. And finally, there's also relative localization, which means that localization on some functions under some condition happens, but they also acknowledge the limitations of localization, which was mentioned in the principle of mass action and equipotentiality. Examples of this would be Gazaniga, Gazaniga 1967 and Sperry 1968. Right, so neural networks is a series of interconnected neurons where synapses connect from one neuron to another, and they carry out a specific function. And these are formed by dendritic branching, which would be explained later. And neural networks can be shaped from neural pruning, and this will also be explained later, and through neuroplasticity. And studies which can be used to support this would be McGarr et al. And Dragunski et al. And many more. All right, so neuroplasticity means that basically it means that the neurons in our brains can be malleable. So basically it can change, and it can increase by the stimulation in the environment. And before that, let's just look back at the definition of neurons and synapses. So neurons are nerve cells that carry electrical impulses, while synapses means that it allows the neuron to send electric or chemical signal to another neuron. So how neuroplasticity works is that when there's a stimulation of a new environment, neurons would continue to fire continually in response to that environment. So when neurons continue to fire up, they would start to create a lot of dendrites, and that's called dendritic branching. And with dendritic branching, that means that there's going to be a lot of synapses available to create behavior, which finally forms a lot of neural networks. And if this doesn't proceed to happen, if this thing doesn't continue to happen, then neural pruning would start happening. So neuroplasticity usually happens in compensation for injury and in response to new changes in the environment. And it's a lifelong ability that will continue and proceed as an adaptational process. So again, the same studies can be applied in neuroplasticity, such as McGarr et al., Dragunski et al., and there's actually also many more, but those are the examples. So neural pruning usually happens when two dendrites are lost. And this happens when the brain thinks that the synaptic connections are no longer needed. For example, in Dragunski et al. 2004, neural pruning happens after participants stop learning juggling. Okay, so for context, Dragunski et al. Is a study about learning juggling, and there was two conditions, which was jugglers and non-jugglers, and the participants of non-jugglers was the control condition, and the jugglers had to learn juggling for 3 months and then stop learning juggling for 3 months. And in every 3-month mark, they would be scanned by the MRI to check on their brain, the brain structural differences. And in this case, because they were learning a skill that they don't usually do, for example, such as juggling, after they stopped learning for 3 months, the structure of the brain who learned juggling the structure of the brain that was involved in learning juggling decreased in size. And this was because they didn't use the juggling skill as much. However, the difference between the person who has learned juggling and who has not learned juggling is still evident. Alright, so now we're jumping into neurotransmission. And neurotransmitter is a chemical substance released at the end of the fiber by the arrival of a nerve impulse and by diffusing across the synapse or junction causes the transfer of impulse to another nerve fiber or a muscle fiber or some other structure. And usually neurotransmitters are stored in the axon and released into the synaptic gap. As you can see here, the axons are inside the nerve fibers, and then when they are released, they're released into the synaptic gap or the gap right there between the two nerve fibers, and after it's released, it would be transferred back into a nerve fiber, a muscle fiber, or other structures. And so there are several types of neurotransmission. There's the oh god, English is so bad. Okay, so there's the, how do you say that? I can't believe I spent my time knowing that word and not spent. Okay, so there's the excitatory increase. There's the excitatory neurotransmission. In this case, it means that it increases the chance of a neuron to fire an action potential. So basically, what this means is that it increases the chance of doing, for example, a behavior in this case. And for inhibitory neurotransmitters, it means that it decreases the chances for a neuron to fire an action potential. And as you can see, some neurotransmitters have can be excitatory and they can also be inhibitory in some circumstances. And on the other hand, so as you may know, usually neurotransmitters in experiments are given to drugs by nasally and etc. And there are two kinds of these kind of substances. The first would be agonist, which increases the effect of the neurotransmitters, and antagonist, which decreases the effects of those neurotransmitters. And then I'll try to cover some of the definitions of neurotransmitters and the examples of the studies one by one. So the first one would be acetylcholine, and as acetylcholine has an important role in cognitive function, especially in memory. Although they also is involved in thought, learning, and memory. So this usually activates muscle action in the body, which is why it can be excitatory sometimes, and it is also associated with action and awakening. So for example, in this study by Rogers and Kistner in 2003, acetylcholine has something to do with memory formation. And because of that, in the experiment, acetylcholine activates the hippocampus to form memory. On the other hand, in the experiment, they used scopolamine, which pretty much stopped acetylcholine from inhibits acetylcholine, which is why it was an antagonist. And then the next neurotransmitter would be serotonin. Serotonin is a contributor for well-being and happiness. That's what it's generally known for. And although it actually has a complex function, such as cognition, reward, learning, memory, and a lot more physiological processes. So, a study on serotonin would be Crockett et al. In 2010 on serotonin on prosocial behavior. So, in this experiment, the neurotransmitter would be excitatory because it increases prosocial behavior. And in the experiment, the thing that was used was citalopram, which is a selective serotonin reuptake inhibitor. Basically, what this means is that it blocks serotonin reuptake, or it prolongs serotonin, and it stops serotonin from going back after it comes out. So the next neurotransmitter would be dopamine. Dopamine, I'm so sorry for the typo, but dopamine helps control the brain's reward and pleasure center. It plays a key role in motivation, and usually when you have low levels of dopamine, it often has links with addictive behavior. An example of a study on dopamine would be Fisher, Aaron, and Brown, which is dopamine on romantic love. In this experiment, they were doing a survey, so there wasn't any pills that they have to take. So the only thing in the experiment was that the neurotransmitters function in the experiment would be excitatory because it is involved in motivation. Alright, so we're now already on hormones and behavior. Do you guys have any questions? If you guys have any questions, you can always put it in the psychology channel, or you guys can like speak up. That's also fine. Okay, but I'll still continue in. Oh. Alright, so the difference between excitatory and agonist is that excitatory is about the neurotransmitter itself. However, for agonist, it's more of for the things that you ingested. For example, in the slide that I have before, you see agonist. When it comes to agonist, it's like the substances that you take in. Well, it does have the same function, but basically the difference is that it enhances the effect even more for agonists. For excitatory, it means that it increases the chances for something like behavior to happen. Or other than behavior, it could be like physiological processes. For example, in adrenaline, it's an excitatory neurotransmitter because when your adrenaline increases, that means that your heartbeat would increase, and then um, you would start feeling prepared for fight and flight responses. So I hope that that clears up your question. All right. So if you guys still have any questions, always always feel free to put it in the Discord channel. So I'll just continue with hormones and behavior. All right. So hormones are body's chemical messengers, and hormones are kind of like similar to neurotransmitters. However, um, in the difference between them is that hormones are merely chemical messengers, and they're secreted by glands in the endocrine system. Unlike neurotransmitters, they're only in the brain, and other than um, a chemical messenger, they're also electrical impulses. So um, for neurotransmitters, things work faster. For hormones, it takes more time. That's why it works slowly. And um, as you can see, it travels in bloodstream to tissues or organs. So it is way slower compared to neurotransmitters. All right, so there's actually a lot of types of hormones, but I'll just cover up the types of hormones that's used in studies. And these are the types of hormones that I'll cover up. And the first would be cortisol. So cortisol is usually secreted in the adrenal glands. Um, it usually is related to stress, and it helps control sugar blood levels, regulate metabolism, and reduce inflammation. It also assists in memory formation. So an example of study to support this would be Newcomer et al. 1999, which is about cortisol on verbal declarative memory. So for serotonin, um, serotonin is a contributor. Again, the same thing as neurotransmitters. It has the same function. Um, an extra side note. So basically, um, there are certain hormones and neurotransmitters that can be the same thing. So one example of this would be serotonin, and then there's also adrenaline, and um, I think if I recall, I think dopamine too. Um, all right, so since we've explained what serotonin is, we'll just jump right into the studies that can be used. One of them would be, again, the same thing, serotonin on pro-social behavior. All right, so for adrenaline, um, just like cortisol, it's also produced in the adrenal glands, and it's well known for its flight or fight response. So how adrenaline usually works is that when the brain sends fear or um, other senses, the amygdala would send a signal to release adrenaline. And after that happens, it will start stimulating the sympathetic nervous system. Um, and after that, there would be increased blood flow to the muscles. And by increasing the blood flow to the muscles, your heart rate and breathing rate would increase. And um, by doing that, it prepares an organism to enter the fight and flight response. And as you can see here, the increase of blood flow to muscles, heart rate, and breathing rate is an example of an excitatory neurotransmitter. And um, so a study that can be used for this would be the role of adrenaline and the amygdala on memory. Okay, so by the way, a quick reminder, um, it's it's very very important that you should know the difference between neurotransmitters and hormones because in most ERQs or SAQs, um, sometimes people don't get points just because they often mistake neurotransmitters for hormones and hormones to neurotransmitters. So it's important to have that difference to keep to make sure that you know that difference and to pay attention of that difference. All right, so let's just continue to oxytocin. So oxytocin is usually produced in the hypothalamus, and it is secreted by the pituitary gland. It controls some human behaviors and social interaction. For example, would be generosity and trust. And it's also known to trigger a bond between the mother and an infant, especially right after, usually when a mother just gave birth. It may also play a role in recognition, sexual arousal, trust, and anxiety. And in some research, they also found that oxytocin can also affect addiction and stress. So, an example of a study that supports oxytocin would be from Gardner et al. It's about oxytocin and the breaches of trust. Right. Pheromones and behavior. So, um, pheromones generally means that it's a chemical that is released by an animal that can affect uh, physiology and behavior of other animals in the species. And usually, this behavior can play a role in reproductive status or sexual attractiveness. In animals, it's usually detected from the vomeronasal organ and the accessory olfactory bulb. In humans, um, the specific pheromone organ has not actually been found yet, but there are several hypothesized are putative human pheromones. And as a reminder, in IB, um, the IB knows that most pheromone most pheromone studies are inconclusive, but usually, even though it's inconclusive, it's important because um, it can help train you in critical thinking and evaluating studies. So, an example of a study which supports um, pheromones and behavior would be vetted by Wettigan et al. In 1995 about smell and partner selection. Okay, so we're already in genetics and behavior. Do you guys have any questions? This is actually going faster than I thought. Alright, so I'll just continue again. Um, okay, so genetics and behavior. Um, so in genetics, um, so genetics is a unit of DNA, and it's located on a chromosome. It controls the development of one or more traits, and it's a basic physical and functional unit of heredity. And its genetic information is usually passed from parents to offspring. And a study which also supports this would be Wettigan et al., which is MHC in partner selection or major histocompatibility in partner selection. So um, it's all in genetics. We also learn about genetic similarities. And genetic similarities happen because, um, as you can know, um, as you may know, human behavior is influenced by nature and nurture. And um, in genetic similarities, several ways to study and compare these kind of similarities would be to study through twin studies, family studies, and adoption studies. So, twin studies usually compare estimates between monozygotic twins and dizygotic twins to concordant rate. And concordant rate means that the probability of twins with shared genes will develop the same disease. And um, so usually in monozygotic twins, they share 100% of the genotype. And this happens because usually these kind of twins came from the same egg, which gets split into two. However, for dizygotic twins, they usually only share 50% of the genotype because they develop from different eggs and they didn't split into two like the monozygotic twins. They come from two different eggs and then like they just happen to get fertilized together. It so if um, usually if identical twins are more similar in behavior or in certain physiological aspects than fraternal twins, usually it means that there are some genetic factors that may take place. So examples of these kind of twin studies would be Kendler et al. In 2016 about moderate depression heritability and Bouchard and McGuey in 1981 about IQ correlations between relatives. So family studies, on the other hand, can't do that because family studies um, is not as close as twin is not as close in genetic means compared to twins. So usually the things that are used would be the degrees of relatedness. Um, and what this means that it compares in the broader scale and across generations. So instead of comparing them um, genetically only, they also compare them by like general, maybe like general interest, and then their general behavior, and etc. An example of a family study would be by Bouchard and McGuey in 1981, again, about IQ correlates between relatives. So the last one would be about adoption studies. Um, Adoption studies compares adopted children to adoptive parents, to biological parents, adoptive siblings, and biological siblings. So usually in these studies, you want to compare between um, nurture versus nature. So you would compare environment, genetics, and see which one has more influence. And one study which supports this or explores about this would be Kendall et al. In 2015 about genetic environment on intelligence of the adopted. Right, so last but not least, um, so humans evolve to survive, or to at least increase the chances of and survive in natural selection. And we do this by passing our genes. And these genes would influence our physiology and behavior, um, which promotes um, adaptation. And an example of this study which supports this would be Redigan et al. In 1995 about MHC in partner selection. So, what happens in the study is that the participants have to, um, the male participants are asked to wear clothes, um, to wear clean clothes when they sleep, and they were also told to like use as less deodorant and become as natural free as possible. And then after 2 nights, um, all the clothes are put into like boxes, and the females would start smelling them and rate the attractiveness and sexiness of the smell. And um, the study shows that most females are attracted to smells that are genetically different from their counterparts. And it's interesting because apparently um, it doesn't matter um, if your genetic variation have the best immune system, because people are literally just looking for genetic variability. And by this, scientists believe that this might have happened because humans are just uh, trying to make sure that their offspring's genes are as variable as possible, so that um, their their offsprings um, can can adapt in many situations. Alright, so the SL content is now done. It's now time for animal research. So anyone here um, who's only taking SL can now leave. Thank you for coming. Wait, are you guys mostly Asia? Oh, okay, never mind. Okay, so let us proceed to animal research. Let us start this by reminding everyone that animal research is not a research method. Don't ever put in animal research as a research method. You can do experiments, you can do quasi experiments, you can do correlational studies, you can do case studies, you can do whatever quantitative study or qualitative study you have. Just don't put in animal research for the sake of yourself and your grades. Okay, now, animal research studies. Um, Animal research is basically a technique of experimenting on animals. Basically, you do experiments on animals like how we do experiments on humans. Um, and usually, this is for biological research on psychology because you're going to have to relate the genetics of other animals to humans and try to generalize it in some way. All right, so animal research have a lot of strengths and limitations. And some strengths would be that human. So since humans and animals are identical in many ways, in brain structure and genetically, this means that if we do experiments on humans, there's a high likelihood that it might be similar in the case of humans, because as you can see, there's a high gene genetic correlation between some humans and some animals. Well, examples would be rats, I think, which has 97% of human genes. And this shows that like rats can be maybe generalizable to humans in some aspects. In so in the past, the reason why some models of behavior came to be is because of animal experimentation. So an example of this would be Harlow et al. In 1965. It's that it's the one well-known experiment where rhesus monkeys. Um, so there's this one experiment how some rhesus monkeys are deprived from and taken away from their mothers, and they were given two choices: a mom with nutritional food and barbed wire, and a fake mom with barbed wire but with nutrition intake, and um, another uh, another fake mom with cloth and horrible food. And interestingly, in this experiment, the monkeys decided to prefer to go to the mom with the warm cloth because um, at the time they found out that um, animals and humans actually need that kind of attention and warmth from parents because in that era humans thought that babies are trying to steal attention to you, so they think that it's not important to give babies attention. um, and because of this study they found out that oh um, having warmth and attention from parents might actually be very important for for babies' development and um, for their future. So, in addition, since some animals have a short lifespan, for example, rats, which has 2 till 3 years of lifespan, it allows researchers to observe their different stages of life, from birth, they're young, they're old, all stages. And it's easier to observe them because, as you can obviously know, we have a long lifespan, and it's hard to observe someone from their birth to death, literally, unlike animals. And the good thing about animals is that you can control them really well because of their requirements. And they're also inexpensive, easy to handle, easy to access, and easy to manage, unlike us. We are very annoying to handle, but that's okay. There are pros and cons. Um, there's okay, so there's also limitations in animal research. One would be that animals and humans aren't exactly the same. So, um, as you can see, in one of the experiments, such as PREMAC 2007, um, humans and animals can have differences psychologically. And so, how they differ psychologically is that in humans, when you learn something, when your parents try to teach you to learn something, it's complex. It's not like animals. You see, in animals, for example, um, in lions, their parents would teach their kids to hunt, to learn and hunt prey. However, in humans, it's not that simple. There are more to it. It's not just I'm going to teach you and you're going to learn and that's it. It's not like that. That's why um, there are some differences. And in addition, um, the bad side to it is that if mice are used to test on drugs, even if it never immediately apply on humans. Um, so like the thing with this is that when mice are used to test on drugs, it doesn't necessarily mean that it would work. And even if it does work, that means that they're going to have to test it on more mice, which would be very unethical um, because they're drugs. Or they have to test it on many other animal species, which would be very daunting. And in addition, animal research can have high ecological validity because usually animal research can be controlled easily, and when it can be controlled too easily, ecological validity can easily happen. An example of this would be mice. The mice is getting caged in a certain area instead of in their natural environment. So when this happens, um, it might be it might stress the mice and it might depress them, which causes them to react differently from their natural behavior. And again, animals and humans are essentially different. An example of this would be um, how HIV vaccines work on most animals but not on humans. On the other hand, aspirin, uh, aspirin is analgesic or painkiller, which is surprisingly dangerous to most animals, but it's perfectly fine for humans. So, um, animal research have several ethics. Um, several ethical considerations. And nowadays, animal study can only be done if they have a clear scientific purpose, um, and if the experiment is effective, and if the experiment is submitted to the ethics committee before conducted, and if the researchers are familiar with the species. So when they say by clear scientific purpose, it means that um, it has to make sure that the experiment has clear use or benefit. And other than that, you have to also make sure that it increases understanding on certain species. So it's like sacrificing for the greater good. Or uh, another another good reason that allows you to do animal study is that if the results that if the experiment that you do might give results that have very good benefits for humans or other animals. And even if you're allowed to do animal studies, you have to make sure that you minimize the use and also make sure that the animals are treated humanly. So the animals aren't treated and degraded just because they're animals. And you have to make sure that using the best species to address research so you're not wasting your time doing animal research. And in addition, researchers have to be familiar with the species, so the researchers can know when the animals are distressed or ensure that the needs of the animals are fulfilled, so that whenever there's distress on the animals, necessary steps can be taken. For example, if there's bad distress on the animals, then euthanasia could be done to relieve the stress from the animals. And um, animals should also be given good welfare. They should live comfortably like humans or better. And animals in lab, if they're used as experiments, shouldn't be released into the wild. Um, and if you've used like a batch of these animals, it's better to use the same batch again instead of using a new batch so that more knowledge can be generated from the same batch. It's either that or you have to minimize your use on the samples. So for animal research strengths and limitations and its ethics, you can always use any of the animal research studies. You can use the studies that I put in, or you can always use the studies that you're going to learn. And that's all for today. Thank you. Thank you for coming. I hope this is very helpful for you. I hope I tried my best.